what does this do? Why is it non-specific?
That's anything that doesn't prove long enough.
Okay. So then what's the third level?
Specific.
Specific.
Response.
And what is this? Specific based on
Type of pathogen.
immunity. So your immune response. Okay. So physical barriers, step one, because we don't, prevent it from getting in. That's great. It gets past the physical barriers, your skin, your mucosal membranes, any of that. Then you have your blood. Circulating through your blood, you have non-specific parts that come, and anything they recognize as foreign, we'll try to destroy. If it can get through that, or it's a bigger problem than your non-specific can handle, then you go to your specific, your immune response. Okay, so this is when you start making your antibodies. So you specifically target whatever is infecting. So, skin. What are some of the chemical factors that help make your skin a barrier?
Salt.
pH, salt,
Heat.
what's one of the other things? What does our salt glands or our sweat glands produce? What's that? How about lysozymes? So enzymes to help kill microbials. Okay, and so we have dermis, epidermis, right? So, and we have epidermal dendritic cells, which just randomly eat whatever, trying to make sure they're not there. Mucosal membranes. What does the mucus do? Why do we have mucus? Okay, so you trap bad stuff. Our cilia is important for sweeping that away, getting rid of that. Also, sometimes if it's trapped in the mucus, then also our body can sample it, know what it is, know how to, maybe prep the immune system for it, or it just kind of goes away. Okay, so also tears. Tears also remove pathogens. They get the dirt and crap out of our eyes. Okay. Plasma. Okay, so blood, non-specific. The liquid portion of our blood is our plasma. So this contains a complement of proteins. This is kind of the basic host defense. Okay, you have cells, you have cell fragments. This is where we get the erythrocytes. What are erythrocytes? Red blood cells. Leukocytes? And then platelets. What are platelets?
Fragments.
Blood flow.
Blood clot. So, have a whole, in our blood, there's a lot of things. Those leukocytes can then be separated out into neutrophils. So, within the broad category of leukocytes, which is your white blood cells, we have these 5, neutrophils, eosinophils, basophils, cells, lymphocytes, and the monocytes, the macrophages, okay? So in general, white blood cells is our immune system. So you're gonna have, the major thing you're gonna wanna do is eat and destroy the bad guys. Okay? So in scientific terms, if a cell is eating another cell, what is it called? Phagocytosis. And so these three macrophages are all part of the phagocytosis. They're all, those three are involved in phagocytosis. They're going to eat something. Specifically, these will go for if you have parasites, but in general, those three classes of leukocytes, their job is to eat something. Okay. Basophils, what are these for? Inflammation, okay. Response. Okay, so it's kind of like you need to get that inflammation going. We talked when we were looking at that earlier. Oh, we were going to look at that video today, but we'll look at it, I guess, Tuesday. But how inflammation helps with wound healing. As a signal to bring more. It triggers. Say, hey, something's wrong. I'm here to help. Lymphocytes. Why are these so important? Okay. These are our B's and T cells. This is our antibody synthesis. Okay, so this is the cell-mediated immunity. Does that make sense?
I'm sorry, but would all of those technically be considered white blood cells?
That's why the white blood cell is not specifically a second line, it can be second or third line. Okay. Questions? Okay, because that's why you have that nice little chart on the next page or slide that tells you about that stem cell, how it can either go into the lymphoid stem cell and become a lymphocyte. That gives you your adaptive immunity versus going into just being a general leukocyte or the innate immunity. So the platelets for clotting, and then all of the other ones other than lymphocytes.
Thanks, Tom.
Most of them are innate, general, and they just kind of chew up whatever they get their hands on. But the lymphocytes, being the T cells, those are very specific cell-mediated immunity for the adaptive immunity. So that's the third line of defense. Make sense? 3 lines. So, phagocytosis. What are the basic steps? What do you have to do?
Chemotaxis.
Yeah, so what is chemotaxis? Why do we have chemotaxis?
Traction of phagocytes.
You gotta have traction, okay? So if there's some kind of foreign pathogen, a microbe, you want the body to recognize this. You want there to be something that says, come here, this is not right, right? So you have chemotaxis. So this is attraction. Then, once it's attracted, what is it attracting anyways?
The phagocytes to the microorganisms.
Usually the macrophages.
Oh.
Those are going to eat everything. Okay. So, attract it, and then what do you need to do next? Once you have the cell that's going to do phagocytosis,
Adhere.
you got to adhere, you got to attach to it somehow. Okay? So, attracted cell, starts to attach, starts spinning out the pseudopod. Then what's going to happen?
It's gonna eat it.
Ingestion, you eat it, engulf it. Okay, now it's inside the cell. Now what? Break it down. Digestion. Okay, you broke it up, took whatever you wanted, now what? Exocytosis, spit it out, eliminate it. So, okay. General, basic pathway, talked about it before, seen it before. Make sense? Then we get to those oh so fun complement cascades. There are 3 types. Classic, alternative, and left. So what is, if we're talking about the complement cascade, what is the general idea of this? What are we talking about? What are these pathways for?
Starting defense. Starting defense.
It's, some kind of starting defense. Because like in the classic pathway, what happens? What makes that one stand out? Antibodies, okay? Classical, we think antibodies. And the alternative, what do we think about?
BDP. Just kidding.
Okay, so the entire point of this, whether it's classical or alternative, is you meet up at that C3. Yeah, so all pathways meet at C3. Okay. So all of them go to C3. They help cleave other proteins. You get the C4s, the C5. They go to inflammation. You get the C5b plus C6, 7, 8, and importantly 9. That all goes to that membrane attack complex. The entire point is to make that membrane attack, put that hole in the membrane of that microorganism. Okay? So, classical, you start with antibodies. So your body already has some antibodies floating around, recognizes the outside of the pathogen, and starts the cascade, with C1, C2, C4 to lead to the C3, which all ultimately leads to the MAC, the membrane attack complex, okay? The alternative pathway is there are some microbial cell wall polypeptides. So there are some things that microbes just have on their cell wall, not familiar to us, but they all seem to have it, and so our body just recognizes that. It's like, those are foreign. We should not allow those to exist, okay? And so, cell wall polypeptides. polysaccharides. Sorry. And right there, that should be a clue, because do we have a cell wall?
Oh,
So, they have a cell wall, they probably shouldn't be in our body. Or a lot of them shouldn't. We do have yeast in us, and those have cell walls, but that at least should be checked out. It has a cell wall, it's not cell. Okay, so this is where we have the factors. B, D, and P. So, alternatively, you can recognize factors B, D, and P, these polysaccharides that are not normal to us, and then that goes, triggers C3, trigger the MHC membrane attack. Okay? Everything goes to making the holes. What about the lectin pathway? So some fungus are good, but we needed a way to make sure some of them that aren't, we know how that we can attack, our body will recognize. And anything your body sees over and over again, it's gonna eventually figure out a way to kind of broad scale, okay, anytime you see this, start this. So you don't have to be specific, you just have to know, I'm under attack, this is the kind of attack, do this. Okay, so lectin pathway, this is where we're going to, it's initiated by the lectin binding to mannose.
I'm sorry, I missed this yesterday on my notes. What is mannose?
Sugar,
Just another form of sugar, another writing sugar.
it's, yeah,
Simple carbohydrate.
it's a form of sugar, yeah. So, three different pathways. The entire purpose is to recognize something is wrong and do that membrane attack complex, okay? So we have, pretty pictures, all the holes that it chops up. Now, when we get, so we kind of, jumped over, one of the outcomes, okay, the outcome is we're gonna get that membrane attack complex, but in the middle there, we're also going to end up getting inflammation. So, the role of inflammation in all this is chemotaxis. It's what's going to stimulate the chemotaxis of leukocytes. So it's going to be that inflammation is what stimulates the signal saying something is wrong. Everybody who fights, come here and fight. That makes sense? It also stimulates the basophils. And this is, all this is a C3A, C4A, and C5. So it's one of those things where you initiate the pathway, break all these proteins in half, and the A's tend to seem to be going and starting inflammation, doing one signal, these going, some of them, what is it, the C5B goes and starts becoming part of that membrane attack complex. So it's one of those things where you need to cascade so that you don't just have, an on-off switch. You kind of need this to bring this, to bring this, to bring this, to bring this. Also, that means that if you take something out, if you kind of decide, okay, we have enough, and you stop it, then it's going to stop breaking everything down, and the cascade's going to stop because you've already, that signal dies, you're not going to make any more, so you just have what you have. So it's a way to have very controlled on and off. interferons. We have type 1 and type 2. Okay, so we have alpha and beta, and we have gamma. So alpha and beta are produced by virus-infected cells. Gamma is produced and activated by T cells. So basically, bottom line is it's going to be stimulated, produced by virus-infected cells, and it stimulates to make antiviral protein. Type 2, gamma, is going to be activated by the T cells, and it's going to stimulate macrophage activity. What do macrophages do?
Eat other cells.
They eat things.
Phagocytosis. So, if cell A gets a virus, what is it going to get? The what?
Cell A has a virus. It recognizes there's foreign DNA coming in. What's going to happen to that cell?
Type 1 alpha and beta immunity, we're going to
Not immunity, but
earn.
so cell A has a virus. So shit, I have a virus. There's nothing you can do for me, but I can turn on type 1 and 2 alpha and beta interferons. And I can send out signals, and I can warn my neighbors. Okay? So they can send out the interferons, type 1, alpha and beta. So this is produced by a virus-infected cell. So cell A is infected, it's going to start producing this. Now cell B is next to cell A. What's it going to do? It's going to recognize the alpha beta interferons,
Start eating them.
and that's going to make it so that it's type 2. So it starts making, it's going to start making that antiviral protein. It's going to recognize that. Okay? So this way, cell next to it has a fighting chance. The original cell is going to die. It has a virus, it knows it has its virus, it tries to at least warn its neighbors. Does that make sense? Then you're also going to have the type 2, the gamma interferons. Once T cells kind of been come along, been activated, going, this is bad, this is wrong, and so it's an active T cell that's actively trying to make our immune system. That's going to start producing the gamma interferon, and that's going to stimulate macrophages. So basically, once the T cell comes, signals, this is bad, this cell needs to die, you then need to bring something in that can actually eat it. Because T cells don't eat cells, they don't do phagocytosis. The macrophages do. T cells are just part of building the antibody immunity. So they might recognize it, they might taste and sample it, but they can't kill it. But they can send signals to get it. Yes.
In this type of type 2 gamma interferon immunity, T cells activate or macrophage activity and phagocytosis comes in, does it just digest the entire damaged cell, or can it attack individual viruses before they get into cells?
I think the size issue, so a lot of times viruses are so small, you won't necessarily, I mean, the cell, you wouldn't necessarily
It
notice
just
it,
cleans up
and
infected
trying
cells.
to attack you. You know, you wouldn't even notice it. So, I mean, it might be able to clean up some of those, but for the most part, it'll
Engulfs
come
infected cells.
and engulf the entire infected cell and try, I mean, because if you can get there before the cell explodes, then you've gotten rid of a whole bunch of viruses. As long as it's a, what kind of virus? Enveloped or non-enveloped? What one lyses? Huh? Non-enveloped cells, they have to lyse the cell so they can get out. Enveloped cells get out by
Version of the cell.
getting out.
Oh,
By budding.
oh,
So yes, they do merge with the cells, get in, potentially. There's multiple vehicles. Merge, drop all their glycoproteins off. But yeah, budding.
right.
Envelope cells, because they're stealing that cell membrane. Okay, inflammation. There are 4 basic symptoms of inflammation. Redness, edema. What is edema?
Swelling.
Swelling.
I hope you know what redness and pain are. Then localized heat. What is the function of inflammation?
So warning.
Destroy the.
Okay, it's to destroy things. You want to try and destroy the harmful agents, so whatever microbial or physical agents or chemical, whatever is there causing an issue, you're trying to destroy it. You're also trying to confine the harmful agents. I mean, if you can swell, then maybe it can't get past you anymore, you know? So you're trying to build some kind of barrier to contain whatever is the issue. Also, it's a nice signal to say, there's an issue here. Repair this. And so a lot of these things are signals to your body to let something else know, to be some kind of signal saying, we have an issue, come help. Okay? So, the steps in inflammation, you get a tissue injury. So you can get vasodilation. What is vasodilation? Diameter increases, okay. You're going to be increasing blood vessel, going to increase. So this is one of those things where you can start breaking apart the organs, okay? So you're going to increase your blood vessel's diameter, and it also increases the permeability of that blood vessel. Okay, so this will cause, it's caused by histamine release, so by the plastophils that we're talking about. Plastophils are part of what part of your blood? White blood cells. Okay. So you end up getting leukotrienes, they increase the permeability of the blood vessels. So you're trying to get this edema accumulation of fluids around the injury, sorry. So the whole idea is that you're trying to build things up, build up the fluids, then you're going to get this accrual of host defense cells. They're all just going to start, gathering at this injury site. Okay? look at your capillaries, what's gonna happen is you're gonna get this blood flow, and all of a sudden, instead of having this little fine net, you're gonna get this really raw net, because all of the diameter is going to be increasing. So you get that dilated capillaries. It also increases that permeability, so you can see a little picture of it that have that nice cell, that nice tube, and then it starts getting, there's space between those cells. And so you can get the monocytes squeezing through the interstitial space. And so they're starting to be able to move, this makes it more fluid, and microbial chemicals can actually leave the blood vessel and get to the site of injury. Because blood does not go everywhere, it goes most places, and then kind of touch on things. So trying to make sure you can accumulate whatever you need for your immune system to start taking care of that infection site. Okay? Just making sense? So you can have two types of inflammation, acute and chronic. So, acute, you got a little injury, injured, it eats up, it inflames, you have the redness, there's some basic pain once you know you did something, and then it goes away. Chronic, you do something a little more substantial, your body's not able to take care of it quite as much, and so it ends up being a long time chronic issue. The acute develops quickly, it's short-lived, it's typically very beneficial because you have hurt your leg, usually you know it, and you stop putting extra weight on it, you give it a little time to rest, and so it's a very good thing. It's our body's way of saying, you messed this up, wait, let's fix it, and then you can proceed. And it's an important second line of defense because you're getting that dilation, it's increasing mobility, it's making it so that your blood is able to get where it needs to actually have all of your immune system get to the infection site. What's the migration of the phagocytosine cells to get there, so they can take care of whatever is there. So you need tissue repair. So, what are some of our steps? We have the phagocytosis. Migration. Phagocytosis. So you want the attachment of these leukocytes, the neutrophils, the monocytes. You want these blood vessels to come show up, and you want them to start eating the bad things.
What else did you say? Develops quickly and what else? Second line of defense?
You pronounce that.
So it's just those two things?
Diabetes?
CBU?
I would say diabetes. Diabetes. Between cells. So, you need attachment of the leukocytes to the blood vessels. So you need to first get the leukocytes there. Then you need to get them to squeeze between the cells and blood vessels so they can actually move to the injury site. And then, now that you have parts of your red blood cells out of blood vessels, they can start actually clotting. What's going on? So you need your clotting factors. So they start to accumulate and they can define that injury. It's usually a good thing, we can stop bleeding. And then you need to do your tissue repair. Something's wrong, tissue is damaged, so this is why we're bringing all leukocytes in the first place, so you can repair that tissue. We showed one video, but we'll show the steps next time we have a computer. Okay. Basic steps of inflammation. Kind of makes sense what it's for. Fever. Why do we get fevers?
I had a quick question. For inflammation, if it's like for acute, it's beneficial. How come they always, like whenever you have an injury, they always put ice on it right away to like stop inflammation?
So, it is technically beneficial, but we like to keep moving and it's going to slow us down. So in a way, putting the ice, you're kind of trying to make a deal with your body going, okay, I know you're injured, don't swell on me so I can still walk, and I promise to be gentle. So it's one of those things where, as far as the body's concerned and healing, it is a beneficial thing because it's protection. I mean, it protects whatever happens. It's like when you have the nerve spasms because something's wrong, and it's trying to guard and protect.
So are you slowing down the healing process by doing that?
By the ice?
Or-
Some you can, but also you basically are preventing some of that inflammation, but already hopefully enough has come through. I mean, because we still heal, but it's more of we're more functional when we do that. Especially, I mean, usually do that a lot with sports injuries because they don't exactly want to be sidelined. So you can kind of get by, but if we just let our body do it and we actually really rest it, would be the best way to take care of those injuries. In general, we don't do that.
Okay.
If we were too impatient, we want to move. Does that make sense? And also, the acute, that quick flush of it, the start of it is great, but sometimes it likes to last a little too long, it starts with that chronic, and then it's not good. It's not being helpful anymore. Okay, fever. What are some advantages to fever?
Increased temperature can kill out whatever it is.
Slows down the microbes.
Okay, so you can increase the body temperature, and so this can be above the optimum for the pathogens, so then it's going to slow down your microbes. Increase. Also, for us, a lot of times, you increase the temperature, you increase the speed. And so, if our chemical reactions happen at a certain rate of speed, at 30 degrees, which is what we're, whatever, then you jump up to 37, it's going to increase even more. And so, we're normally at 37, you start jumping it up to at 40 or whatever, trying to get towards that 100-degree Fahrenheit mark, you're going to start increasing those chemical reactions. There's a number of fever signals. So things that result in a fever, microbial production of some of their exogenous start making things, your body might go, this is bad, let's try and slow this down. Phagocytosin releasing the endopyrogens like interleukin-1, so all of a sudden, when that macrophage gets there, it eats the cell that was full of virus, and it goes, there was an interleukin here, we need to deal with this. And so if it raises the entire temperature of the body, then you can start trying to fix this, okay? So, microbial products. You can have things that they start triggering your hypothalamus to release prostaglandins and some of those things where there's a lot of little triggers that let your body know we're under attack. We need a system-wide defense. Because that's what a fever is. It's not specific inflammation, which is site-wide. This is system-wide. So it's kind of that difference of how big of an infection is this? So prostaglandins will reset our hypothalamus at a higher temperature, which results in fever. Understanding of fever is helpful. Hypothalamus is secreted. It's all due to your hypothalamus.
So it's the interleukin.
But some of the unpleasant side effects of fever, as we all are familiar with, generally just don't feel good. Body kind of aches, tired. It takes a lot of energy to raise our body temperature. That's why we eat food, is to keep ourselves at a constant temperature. If you're now having to ramp that up, that means it's taking more energy to do that. And generally when you're sick, you don't really feel like eating more, so you're just kind of taking on reserves you don't have, so then you don't have energy to do anything else. Okay?
Can it also damage